Welcome to the first thematic session of the 2020 CLIFF-GRADS webinar. So I'm Siniro Costa Jr. I work for CCAF's flagship tree on low emissions development. And today I will be facilitating or chairing, as you will, Todd, today's session that is focused on integrated and agroforestry systems. So to recap a little bit about thematic sessions. So these thematic sessions, they have been designed to provide students the opportunity to present and discuss their PhD research with colleagues and the guest expert. There will be 4 more thematic sessions covering pastoral and agronomy, ruminant, rice, and soil. And today, we are going to have 5 students, CLIFF-GRADS students presentation, followed by discussions. Today, this session will be chaired or facilitated by Todd Rosenstock. So Todd will introduce himself later on. And before we start, let me only say how we are going to manage today, or how we intend to have this session today. So first, Todd will give us a 5-minute introduction, and then we are going to have 5-minute student presentations. There will be recordings, and then we are going to move to a questions and clarification and comment session. So if you have a comment or a question or want to clarify some point, please add that in the chat bar. So you find the chat bar here in the lower right hand side of this Zoom meeting, right? And then after these presentations, we can read out your question and then we start discussing it. So Todd, again, thank you so much for being here with us. And I turn that over to you. Thank you so much.
Thanks, Siniro. One point of clarification right at the start. We'll have clarifying questions between each individual student's presentation. So if you have a specific comment about why did you use a vent on your chamber, or what kind of allometric model you use for this species, that's the appropriate time to ask that. When you're talking about broader, more general questions that are for discussion, then we would save those for the end after we've seen all 5 student presentations. So there's 2 opportunities, and of course, always feel free to enter information, chats, things that you're thinking into the chat box. It's an active area where you can interact despite the presentation that's going on, despite while I'm talking, whatever it is. Just have a conversation amongst yourself. I don't understand this. I don't believe this. This is totally fine. And that's an active way to participate with all your other colleagues and students that are on the chat. So I'm honored to be here today. I have to say that I was one of the first people to have started the CLIFF-GRADS program with Meryl Richards and others many years ago, 5-6 years ago, and I've lost a little bit of touch with the program. So I was very excited when I was invited here to talk today. I wasn't quite sure why, but I'm very excited to talk. So my name is Todd Rosenstock. I'm a senior scientist at World Agroforestry Center. And so normally I'm based in Nairobi, Kenya. I've been living in either Nairobi or the Congo for the past 10 years, where my work is largely focused on climate change mitigation and climate smart agriculture. And more frequently right now veers even further towards the adaptation. So, and you'll see some of that probably in some of what I'm going to say. I was only asked to give 5 minutes. I think I've probably used 2 minutes already, but I think I'll ask Hazel for chair's prerogative, and I'll go perhaps a little bit longer. Let me share my screen and just give a very brief introduction to some of the things that I think about when I think about these issues. Okay, PowerPoint. So I'm here to talk to you today, and thanks for the opportunity for 5 minutes about agroforestry and integrated systems. I'm really, over the next 5 minutes, I'm going to try to set my timer to make sure I know that I'm approximately on time. Over the next 5 minutes, I'm going to try to convince you that really integrated systems are everywhere. At the same time, they're nowhere. This means, and because they're integrated systems, they're much more complex than the sort of more simplified, straightforward systems that people are largely doing greenhouse gas work on, as well as, but this all opens new opportunities, and those new opportunities lead you and hopefully provide you a platform for innovative science and innovative impact in this area. And so I really see this as one of the most exciting areas to do climate change mitigation work on. And so I hope this this talk inspires you to start to work on these types of systems in the future. Come on. Okay, so as I said, agroforestry and integrated systems are everywhere. And why would I say that? Because I would argue that the most iconic systems globally, when you go outside into rural areas, are agroforestry and integrated systems. Think about the tree crop systems, the parklands of West Africa that cover hundreds of thousands of hectares. Think about the silver pastoral systems of Latin America that are across Brazil, Colombia, the Amazon, and everywhere basically. And so and you can go on and on. These are the rubber systems of Southeast Asia. Think about the landscapes where you work. And when you stand out there, almost everyone, unless you're in the Midwestern United States, you see trees and you see cows and crops and everything mixed together. And so these really are, even when people are saying, sorry, I'm in the sun, even when people are working on more simplified systems, they're often just working on parts of the system. So they're not really working on systems at all. They're working on specific problems that are part of broader systems. And it's a real challenge to work at this sort of systems level because there's so many different things occurring. But why do I think, even though they're everywhere, I would also contend that they're nowhere? So we did an analysis of the naturally determined contributions. So the blueprints that countries applied for the put in for the UNFCCC for their climate action. So if they named, in this case, it was agroforestry in these NDCs, either for mitigation or adaptation. What we found is in 148 non-Annex 1 countries, so this is mostly the developing world, 40% had named agroforestry. They said 40% of the countries globally said agroforestry is going to be part of our solution for mitigation and adaptation. And guess what? Almost none of them actually include them in their greenhouse gas inventory. So they're not actually counting their benefits. So although everybody is talking about these agroforestry and integrated systems, nobody is actually doing anything about them. And the way that they're measuring this and the way that they're starting to act on it is actually not occurring. And what this means is that there's this huge opportunity for you to have impact and for you to get information into these international policy arenas when you work on these systems. A lot of people are working on enteric fermentation. A lot of people are working on soil carbon. Very few people are working on these systems. But when you start thinking about systems, as I said, it starts to get complicated. You know, this is a picture from Western Kenya, and this is in a landscape that I worked in for many years. And what you have is these mixed systems. You have cows that are producing methane. There's pasture and carbon sequestration happening. There's cash crops, that's tea in the background on the left, that they're using fertilizer on. So you're seeing nitrous oxide emissions. But emissions are only part of the story. The farmers in this location don't care about emissions much at all, but what they really care about is income diversification, use cattle for insurance for when shocks happen for their lives or somebody dies, etc. And so when you think about systems, and even when you're thinking about emissions, in systems, you have to start thinking about things beyond just the emissions themselves. And this is where I think it starts to get really interesting, and where you start to see these overlaps between mitigation and complementarities between mitigation and adaptation. And what this drives you from is, I think what we'll hear a lot about is about field measurements of emissions, field measurements of sequestration. But we know that greenhouse gas accounting, so that second slide that I showed you about the greenhouse gas inventories, is essentially a function of two different things. That's emissions factors and activity data. And there's been a huge push over the last 10 years for better emissions factors, especially in the global south. much less information about the activity data. What are people actually doing on their farm? And guess what? There's a lot of uncertainty around that. We don't know what farmers in Zambia are really doing on their farm. We have very little information on that, and that could completely throw off what we know about agricultural greenhouse gas emissions. And so when you widen the problem beyond just the simple measurement problems, you end up looking at all kinds of different types of approaches, some of which I imagine some of which are very applicable for this integrated systems accounting. And that's remote sensing, aerial photography, even crowdsourcing. So sending people text messages and getting information back. And what this would allow you to do is actually be able to unlock the finance needed for massive transformation at scale. And so I guess I was sort of challenged when trying to put together this presentation, because we have 5 talks that I think get to many pieces of this sort of framework that I've just outlined. And I guess I would challenge all of the CLIFF grads, not the ones on here, and hopefully the ones that will watch this at home as I think it's being recorded, and to think about how their work, although it's just one piece of broader systems and broader sort of political economy and science on greenhouse emissions, can lead towards this transformative impact and this transforming science going forward. And with that, I think let's go and let's get started. Thank you, Suniro.
Thanks a lot, Todd. That's a great presentation. Thank you so much. So now I think Hazel will play out the recordings for 5 presentations in a row, I guess.
Greetings, my dear CLIFF-GRADS mates. Greetings to my supervisors. I am Mary Ngaiwi from Cameroon, and my host institution is CIAT, Cali, Colombia. My PhD is on climate-smart agriculture, climate change, and livelihood security. Rising fossil fuel burning and land use changes have emitted and continue to emit increasing quantities of greenhouse gases into the Earth's atmosphere. Example of these gases, we have carbon dioxide, methane, and nitrous oxide. A rise in these gases equally causes a rise in the amount of heat from the sun withheld in the Earth's atmosphere. Normally, heat that is supposed to be expelled, like to be sent, radiated back to space, is now retained in the atmosphere, which is the main reason behind the greenhouse gas effect and subsequently climate change. The ability to contain the pace of this phenomenon by keeping the change of temperature at the 20 degrees centigrade threshold as stipulated by IPCC 2014 is now limiting, and the entire universe has to suffer these consequences. In this light, the correlation between agriculture and climate change still stands distinct, mostly because of agriculture's impact on global food security and production. We have 4 objectives in this study. First is to determine the farmer's perception on climate variability, and we will need data on perception on rainfall, temperature, age, gender, household size, religion, month per capita income, primary occupation, educational status, and land ownership. The method to be used here will be descriptive analysis and binary logistic regression. Secondly, we will have to look at identify the climate smart practices in this area, and we'll need data on farming systems, adaptation strategies, mitigation strategies, and the types of crops cultivated. We will use the PCA via PAST. And thirdly, we'll analyze the socioeconomic determinants of farmers' adoption of these practices. We have similar data with objective 1, and here we'll be using multinomial logistic regression model. We'll also evaluate the effect of these practices on livelihoods, where we'll need data on farm income and food security. And here we'll use the multinomial endogenous switching regression to capture that. Now, we'll have data at the end on climate smart practices used in this area or practice in this area. We'll also have data on the factors limiting the adoption of these practices, not leaving out the gender aspect. We'll also have data. We'll also have information on their perception on climate variability, because the way I perceive climate is not the same way somebody from another place will perceive climate. I might even be perceiving in my area where I am, and that person is not perceiving yet in that area. So we'll have information on their perception on climate variability. We'll also have information on those practices that will impact them positively and will impact them negatively. So at the end we'll be able to propose to them which one to adopt and which one is not advisable. Now, most of the countries in Sub-Saharan Africa, including Cameroon, rely on agriculture for their income and food security. And any initiative that might help to sustain and improve productivity in agriculture would be a crucial step in improving people's livelihoods. Thus, adoption of these practices is a key step in reducing the threat of the sustainability of agricultural production in Cameroon. Climate-smart agriculture normally is a relatively new concept which was launched in 2009, advocating for better integration of adaptation and mitigation actions in agriculture in order to capture synergies between them and to support sustainable agricultural development for food security under climate change. This is because the relationship between climate change and agriculture is a two-way something. Climate change impacting on agriculture and agriculture subsequently causing climate change. So these methods were developed in order to curb that and to be able to ensure sustainable production and to improve on food insecurity. Thank you for listening.
Great, thanks, Mary. Really interesting. Anybody have any clarification comments before we go to the next presentation? Either speak up, raise your hand, or type them in the chat. Anyone? I have a question then. Mary, how are you gonna document who has adopted and who has not adopted? Oftentimes, in this part of Cameroon, as I understand it, and across much of sub-Saharan Africa, you get partial adoption of CSA agricultural practices. How are you determining that?
Okay, actually I've collected data already. So how we captured it, we did a household survey. And in these areas, we had a list of these practices from intercropping, agroforestry, and the others. So the farmer was able to indicate which one they are using in their farms. So that's how I'm still working on the data. But from the preliminary analysis that I've tried to do to look at how the data is, and there are some practices actually that are specific. Like we had two regions, and we chose two regions. We selected two regions in these two divisions in these regions each. So we realized, like from the data, when I was doing the preliminary analysis, I realized that there are some particular practices that will be for a particular division, probably because of the landscape, the environmental, other environmental aspects, and also with the gender rules and land tenure issues of that area, because it really impacted on their adopting and on their choosing a particular practice. So what we did is we had a list of these practices where we explain to farmers what kind of do you actually practice this particular. We try to explain to them so they understand. Some instances they say, yes, with these reasons I've not been able to do this on my farm because of this reason, and that reason. So that's how I intend to capture which of these practices.
Great. I think that leads into two questions that came up on the chat that directly relate to what you just said. The first one is, how big is your sample size? How many farmers did you sample? And then and how were they chosen?
Okay, the sample size for the two regions was 400. I did a sampling. At the beginning I used a formula for calculating sample size for an unknown population, because I could not get the population of these regions. Since Cameroon has issues on population data, it's like they don't, it's been long that they did the census, so I can't really have an accurate data for the population of this region. So what I did was I used the formula to calculate sample sizes of unknown populations. And now how I selected probably the household, I think that's what they mean.
Yes, I think they're asking what kind of procedure did you use to select the households?
Okay, we did, there are some areas at the beginning, what we proposed, we had to do, how do they, random, is it random? How do they call this?
Random,
Please let me just
random
explain.
is the way.
Maybe I'm a bit tensed. Yes. So, yes, but when we reach, there are some villages, because in each region we had 2 divisions, and in each division we had each village per subdivision. So now what happened was that we did random for some villages, but some villages we realized that they have this cluster. Like the houses were in clusters. So we had to do the cluster, like from each cluster. In each village, we had 20 households. So when we get to the villages that the houses were in clusters, we count the number of clusters. And then we now distribute how many households we visit per cluster. And now when we go to a cluster, we do random amongst the cluster.
Great, thanks. I think that's a good example
Yes.
for everybody who's doing household surveys, that oftentimes you can't do it exactly. She brought up 2 key points that you wouldn't have necessarily expected when you were setting up the research questions. First,
Yes.
you don't know how many people are there, so you don't know your population, and then also this clustering issue in household selection. And so both of these things will affect your research, and something to consider.
Hello, my name is Fernanda Leite. I'm from Brazil, and my CLIFF-GRADS host organization is in Intabalcars, located in Argentina. My PhD title is Soil Organic Matter Quality and Potential Mitigation of Greenhouse Gases in Integration Crop Livestock Forestry in the Brazilian Cerrado biome. Brazil signed the Paris Agreement from COP21, and the NDC, the National Determinant Contribution for Brazil, is to reduce 37% of the emissions until 2025 and 43% of the emissions until 2030, and this based on the emissions from 2005. So for all the sectors, the Brazilian government created plans to achieve this goal. And for the agricultural sector, the government created the plan of low carbon agriculture. That's a plan that create technologies to develop a more sustainable agriculture. And one of the technology is the integrated crop livestock forest systems, and created this network, the ECLF network, to implement this technology and to increase the areas from the agricultural areas with this technology. Nowadays, the Brazil has 15 million hectares with this technology. So this technology, integrated crop livestock forest, has in the same area 3 components: annual crops, pastures with animals, and forestry. So both of the components can have benefits between each other, as nutrient cycling and protect the soil quality and carbon storage in trees and in the soil. So my global objective from my PhD thesis is to access the quantity and quality of soil organic matter and greenhouse gas flows in the integration crop livestock forest systems with different tree densities and ages of the implementation in the Cerrado biome. So my specific objectives are to determine soil organic carbon stock in the ECLF systems, evaluate soil nitrous oxide and methane flows from soil, and determine the organic functional groups of the soil organic matter. My field experiment is located at Embrapa, that's Agriculture Brazilian Research Corporation. In this experiment, it has 5 systems with different ages of implementation and densities of trees. And we're going to open trenches in each system where we are going to collect in the format samples to access the book density and the format samples to access the carbon content. And other analyses for the soil quality made from these trenches too. The gas from the soil will be accessed by the static chamber methodology, where we bury the bases from the chamber and close it with the top. And then with a syringe, we take the samples and analyze in a gas chromatographer. Then we can have the flux, and then we can calculate the accumulated emission in the period. The soil organic matter quality will be analyzed by the nuclear magnetic resonance from carbon-13 spectroscopy and Fourier transformed infrared spectroscopy, that we can see the functional groups of the molecule. To finish, the integration crop livestock forest system is a technology that increases the production and profitability compared to a monoculture and ensure food security. So decrease deforestation because you are using areas that are already open. You can recover degraded areas with this technology. So we don't need to open new areas to implement this technology. And the best one, decrease the greenhouse gas balance. So with this technology, we see that produce and preserve is possible. And this technology is increasing a lot in Brazil, and that's what I'm going to study.
And perhaps we can take advantage of this moment to take clarifying questions from Fernanda's presentation that we jumped. So Fernanda, would you like to make a question, a comment, or a clarification? Anybody has a point for Fernanda's presentation? We have some questions in the chat. So questions from Fernanda. How are you going to take care of spatial distribution of carbon within the farm where livestock graze and congregate, water points and pens?
Yes, we are going to take care about it. We are going to collect to make a transect. In the presentation, I didn't have time to say about these details, but we are going to open trenches near trees and enter the rows, enter the tree rows in the crops or in the pastures. So we are going to take care about these degrees of, we need to see what's the difference in the soil or in the gases. We're going to collect samples from soil and gases in a lot of distance from trees, near trees or in the middle of the interrows.
Great, and there's another question that, could you kindly explain the significance of the functional groups?
Yes, it's organic functional groups is from the organic matter. We need to access this because it explains how is the structure of the organic matter. So we can see if it's liable or it's more refractory with as with lignin and which kind of carbon is there. So we can see, because one of the stabilization process is the biochemistry of the carbon. So we need to see these functional groups of the soil organic matter molecules.
I have a question for Fernanda.
Yes,
Please
Sarah.
go ahead, Sarah.
Fernanda, I would like to know, I'm very interested in the quantification of nitrous oxide. So maybe if you can deepen more in the, how are you going to take the samples for the quantification of nitrous oxide? So how many samples are you going to take in one day, in which hours of the day, or maybe deepen a little bit more there?
Yes. So the best time we have a work here in Brazil is to collect between 8 and 10 a.m. Right in the morning. We collect 4 samples because I hear just the final of your answer. Can you repeat the full? Because it's broke the connection. I don't know what happened. You want to know the time of the collection? And
yeah, the times and how many samples through the day.
Yeah. So the best one, the best practice to take 4 samples per day, because you can have a more accurate calculate of the flux. But you cannot do this. You can collect 3. So, but the best one is collect 4. There are people that collect 2, but the best one is to collect 4. So we are going to try to collect 4 samples per day. In our management is to collect 4 samples.
Okay, thank you.
Just to clarify, Fernanda, nobody should collect 2 samples and estimate a flux. That's bad practice, and so we should make sure that people know that. In terms of, and just, you're talking about 4 samples over the course of a half hour or an hour, right? Or you're talking about over the course of the whole day?
No, just during the deployment period, like 30 minutes or 40 minutes.
Perfect. I think I just wanted to make sure that that was what you were talking about. That's what I thought.
Yes.
But I wanted. Yeah.
Yes.
And the GRA, Sarah, the GRA has put out some very good nitrous oxide chamber methodology protocols. So if you don't have those, there's a number of them out there from different organizations, but I think the most recent and one of the best is really the GRA, who's funding this CLIFF-GRADS. So you should definitely look at those. It goes into all of these issues about deployment times, diurnal variation, fluxes, vents, and all of these very specific things that can have a very big impact on your flux measurements. So please check them out. And if not, feel free to email me and I'll point them. I'll send them to you.
Thanks, Todd. I'll look for them. Thanks.
Can I also come in, Fernanda?
Please.
Okay, maybe to help on how we can work on the number of samples to take. Sometimes we might need to pre-sample and do the linear regression, see whether these need to take 2 or 5 samples. Maybe the emissions are very low, so that just taking 2 samples is okay, because the maybe the fluxes are linear. We're talking of maybe like in sub-Saharan Africa, we're talking of very low fluxes, so that you cannot go to even 5 samples. So it means you can do a pre-sampling so that you can really see whether there is a linear kind of a flux or not, so that it warrants you taking 5 samples, you taking 3 samples or so. Thanks.
It's a question for me.
Yeah, Fernanda, it's just a comment, kind of.
Okay. Sorry. Okay.
Yeah, I'd welcome for you to send me the research that shows that, well, we need to be very careful about flux measurements, especially of trace gases, are I agree can be very low, especially in sub-Saharan Africa. And so, but drawing conclusions from 2 points is not good practice. So 3 points can be done. 2 points is not good practice.
Hello everyone, I am Sara Valencia from Colombia, and I'm currently doing my PhD on Science in Ecology and Sustainable Development in Mexico at the College of the Southern Border. This is a research center that is part of the system of public research centers of the National Council of Science and Technology of Mexico. My host organization is the International Center of Tropical Agriculture at Palmira, Colombia, and the title of my thesis is Use and Enteric Methane Mitigation Potential of Forest Species of the Mexican Tropic. Extensive cattle production systems in tropical regions have generated severe impact on soil and ecosystems and continue to promote land use change. These are also less efficient due to the low quality of the pastures, poor management, and its dependence on external inputs. Additionally, these systems have a higher contribution to greenhouse gas emissions. However, tropical regions worldwide have a high forest biomass production from trees, shrubs, and herbaceous plants with high nutritional quality, with the potential to be used as suitable feed alternative in sustainable livestock system and reduce methane emissions. The Lacandon rainforest is located to the east and northeast of the state of Chiapas in southern Mexico. One of the main economic activities of the indigenous peasant of this region is extensive cattle production and has generated the greatest changes in land use in the history of the Lacandon rainforest. The biodiversity of this region is one of the most important in the country. This characteristic allows a reconversion of the livestock towards sustainable systems through the use of native resources that have the potential to reduce methane emissions and rehabilitate degraded areas. The general objective is to identify forest species little studied in the southern Mexico and to evaluate their effect on the synthesis of methane in the rumen, nutritional quality, effect on animal behavior, and their potential in tropical livestock systems. Some of the specific objectives are to identify multiple use species and their capacity to mitigate enteric methane as an additional ecosystem service based on the information from previous studies, to quantify the nutritional value content of secondary metabolites, measurement of kinetic fermentation parameters, and in vitro methane production of forage species, and to evaluate the effect of diets with forage species containing secondary metabolites on enteric methane production measured in open circuit respiration chambers and rumen microbiome of cattle. In the first experiment, enteric methane mitigation potential of forage species from southern Mexico were evaluated. The samples were taken in the municipality of Ocosingo Valley that covers the largest region of the Lacandon rainforest. The species used for the study were selected based on local knowledge and cultural importance for livestock producers. A total of 15 species were collected. Chemical analysis and secondary metabolite quantifications were carried out. In vitro digestibility and in vitro gas production technique was used to quantify methane production by chromatography at 12, 24, and 48 hours of incubation. In the second experiment, enteric methane mitigation potential of 3 species from tropical regions supplemented at 2 levels on a grass-based diet was evaluated. 5 species were selected from the previous experiment and evaluated at 15 and 30% mixed with a pasture. Also, in vitro gas production technique was used to quantify methane production in this experiment. In the third experiment, the evaluation of the effect of 2 forest species from southern Mexico on enteric methane production and rumen microbiome will be carried out. This experiment was programmed by April, but due to COVID-19, it has been delayed, and we're hoping to start by the end of September. So in this experiment, 3 different diets will be evaluated in a crossover square design where 6 cannulated heifers will be used. Methane production will be measured in 2 open circuit respiration chambers. There will be other variables that will be measured. And for this experiment, my CLIFF-GRADS supervisor contacted investigators from Tiagask in Ireland, so hopefully I will have the opportunity to carry out DNA extraction and sequencing from rumen samples of this experiment there. These are the species that have been evaluated in each experiment and the ones that will be evaluated in the last one. The expected outcomes are select promising species for enteric methane mitigation in tropical systems of southern Mexico, verify the mitigation potential of native species evaluated in vitro in an in vivo trial using the gold standard method for the quantification of enteric methane, advancing the knowledge of the ruminal microbiome by quantifying the effect of secondary plant metabolites on ruminal population and their contribution to methane synthesis. Motivate research and evaluation of the use of native species for greenhouse gas mitigation and animal production in tropical regions. Thank you for your attention.
Thank you very much. Any clarifying questions? I guess my first clarifying question is, how are you going to do all that during a PhD? That's a serious amount of work.
Well, I already started. The 2 first in vitro experiments, we already made them. So we have something advanced there. And the second one, well, the last experiment is the one that I'm worried about because I was going to do it starting this year, but due to COVID-19, we're gonna have to start by September, I think. And that's a long experiment, but I hope I can make it.
Yeah. Other questions? There's a question in the chat, Sarah. The palatability of those materials for animals?
Okay, those species were selected from previous studies. In those studies, they evaluated the intake of these species in cattle, but they were made through the observation of the livestock producers. So some of these producers from these regions, they use secondary vegetation, like in dry season, some of them, not all, and they take the cattle to pasture in secondary vegetation, and they have been observing through many years what cattle are actually eating on that secondary vegetation. So from those studies, I selected those species.
So really important to use things that farmers want. So nice job.
Yes, we are considering like cultural importance and all of that to select those species.
Great. Great, thank you very much. Saniro, is the next presentation queued up?
Thank you, our sponsors, for the opportunity given. My name is Lawal Abdul Hakim Ahmed. I'm a Nigerian. My host organization and country is Soil Research Institute, Ashanti, Ghana. My PhD research topic is vascular plant diversity and above ground plant biomass in relation to land use type in Jigawa State, Nigeria. Savanna ecosystems are being degraded by land use change and other environmental factors, thereby exposing numerous vascular plant species to vulnerability. This has actually contributed to loss of many indigenous vascular species and influenced lots of environmental problems in northern Nigeria. Northwest Nigeria is characterized with average small diameter trees, shrubs, and then grasses that serve environmental, recreational, and medicinal functions. However, the continued existence of these valuable resources in Jigawa State is at risk of loss due to annual bushfire, soil compaction as a result of overgrazing, slash and burn agriculture, or controlled usage of agrochemicals, and then foliar extractions. There is a mutual relationship between soil, vascular plant diversity, and the biomass accumulation, hence the following specific objectives. One, to assess vascular plant species composition and distribution in different land use types, to determine the relationship between soil nutrient composition and woody species diversity across the land use types, to determine the effect of land use on above ground plant biomass, and lastly, to assess the effect of land use on the socioeconomic livelihoods of the people in the study area. Now, this is the study area description. The major land use here are crop farm, agroforestry plots, grazing routes, fallow lands, and then protected areas. These are the land use types on which this study is going to focus on. Sampling procedure. 500 by 100 meter sample plots with 3 replicates will be randomly established on each of the land use types. Within each 1 hectare plot, 1 5 by 5 meter subplots will be located at each of the 4 corners and in the center, while 1 meter by 1 meter microplots would also be allocated on each of the 4 corners at this and at the center of each 5 meter subplot as been depicted on the sample plots layout. Now, 3 species would be investigated within the 100 by 100 meter sample plots, while shrubs and then saplings would only be restricted for investigation within the subplots. Herbaceous species and then seedlings would be investigated within the mini plots, that's the 1 meter by 1 meter plots, mini plots. Now, objective 2. 100 by 100 meter sample plots would be randomly selected of each of the land use types and then will be divided into 3 equal parts, from which soil samples will be collected using a soil core sampler at 3 different depths with their respective replicates. Objective 3. 2 subplots will be randomly selected at each of the land use type for the above ground plant biomass estimation. All woody species with diameter at stump height greater than or equal to 5 cm within the 2 subplots will be estimated using indirect biomass estimation. Questionnaire will be administered in the case of objective 4. Now, data analysis. Objective 1 will be estimated and then presented in tables, charts, such that it will reflect density, frequency, important value index, family important value index, and some other ecological parameters, after which one-way analysis of variance will be used to test for the differences in mean values of our 3 species composition across the different land use types. Now, for objective 2, soil physical properties will be analyzed to determine soil total porosity and then water holding capacity. Soil particle size, texture, and some selected chemical properties will be analyzed in the laboratory, while the data generated will be subjected to PCA. Objective 3, indirect biomass allometric equation model developed by Shape et al. Will be used to estimate 3 biomass, and then one analysis of variance would also be used here, and then least square will be applicable where needed. Chi-square will be used to run the analysis in the case of objective 4. Output. The effect of different land use types on the composition and distribution of woody species will be investigated. The relationship between selected soil nutrient composition and woody species diversity across land use types will be established. The outcome of this work would allow us to identify the best land use options in terms of our biomass stock for sustainable woody species diversity management in Jigawa State. Lastly, the effect of land use on the socio-economic livelihoods of the people in the study area will be investigated. Thank you for listening.
Great, thank you, Abdul Hakim. Any clarifying questions? I feel like I saw some in the chat. Did they want to raise them themselves? Armwell? One of the questions was, what are the land use types? And that you are looking at, and I think the land use type should be in the specific objective so that it is clear. Seems more of a comment, but Abdul Hakim, did you want to expand on this?
That if the land use types could be embedded in the objectives?
Yes, they're just clear, more, they're clear in the presentation. And I think it's more of a comment. Any other clarifying questions? Siniro, any clarifying question?
No, not from my side.
Other questions, comments? We saw a lot of different suggestions of methods being presented today. We saw spectroscopy, we saw respiration chambers. Any comments that can help lead into your own research? Sebastian? You're muted if you're trying to say something.
Oh, sorry. I was thinking about methodology. And yeah, I wonder if someone of the presenters is going to talk about how the methodology is adapted to the assumed linearity or non-linearity of the flux. So that is an important topic to define the number of samples that we are going to take per chamber. Because sometimes in soils that are not frequently annegated or flooded, perhaps the flux tend to be more linear than in other kind of soils, or also depends of the design of the chamber. That's why I am very glad that the GRA has published this last update on the protocols.
Did anybody like to respond to Sebastian? We heard many people talking about fluxes. Fernanda? Okay. Anyone else have any comments on methods? So with Sebastian, we heard another call to read the protocols. So this is great to see the synergies between the GRA research arm directly informing the CLIFF-GRADS research. This is great. I have to say I use the old ones quite often in my own research. Other comments? We also saw some people, there was a previous question about endogenous switching. Mary, did you want to talk a little bit about what endogenous switching is and why people use it? Mary, maybe Mary's not there. Any other ideas?
I can see Mary. I've just requested that she unmute, but.
One thing we didn't see, well, we'll just, if she comes back, we'll go with her. One thing we didn't see a lot about was woody biomass. When we start talking about integrated systems, we heard about the pecans, and I think it was Fernanda's experiment, but we didn't actually see people talking about measuring woody biomass. And there's a lot of conversion of forests and a lot of loss of carbon in woody biomass, and there's also a lot of carbon stored and accumulated in biomass. And so, is anybody working on projects with woody biomass and measuring woody biomass on this group? Sebastian, if you're trying to speak again, it looks like you're still unmuted.
No, sorry,
Okay.
because when you mentioned biomass, I was thinking in another methodological aspect, that is the biomass growing inside the chamber when you are trying to evaluate emissions in an integrative system. Because you know the grass there is always growing, so you have to control that. And there are different approaches to that issue. And sometimes people just cut that grass growing there. And there are other people like supporting the chamber with another ring or another base. You know, I mean, changing the volume of the chamber during the sampling. So I wonder always which one is the best approach to solve that issue.
Is this a question that you'd like feedback on? Or do other people have comments on what they think is the best approach? Suniro, do you have a comment on what you think is the best approach?
Well, yeah, we know that the volume of the chamber influences the fluxes calculation, right? And of course, if the biomass that is growing inside a chamber should be accounted if this biomass is significant, right? Of course, we need to take it into consideration. But depending on, well, and there's also the issue of the length of the experiment, right? So, but my first reaction is that I don't think we should bother on the grass that is growing inside of a chamber, because the biomass can be relatively low, right? Depending on the size of the chamber you have. But Todd, any thoughts on how to proceed in terms of calculating the volume, or should we mow this grass inside the chamber? What is the recommendation?
Yeah, in all of my experiments, we cut the grass, mostly because we were thinking about soil flux and only, and so we ended up cutting the grass.
Great. And another, yeah.
Armwell.
Go ahead, please, Shumba. Go ahead.
Yeah, my take on this is that it depends on the objective, whether it's soil emission, or you want to also maybe take care of crops emission, but mainly it will be carbon dioxide. So if we're dealing with soil emission, like Todi said, you just have to cut the grass. So it depends on the objective that you're working on. So that's my thinking. That's my take on this.
100% agree with that, and
yeah.
So, and I think that's what you would see in most of the documents as well.
Yeah, and
it
going
like,
back to the
sorry, go ahead, Suniro.
To the original Todd's question on how you are measuring woody biomass, right? And I wonder how Fernanda is doing to measure the pecan growth along her experiment. So is there any, what is the methodology, Fernanda, you are using to measure these woody biomass growth?
So in my thesis, I will not collect this data. I'm going to collect just soil for see the structure of the organic matter and the soil organic carbon, and the quality of the soil organic matter, and the soil fluxes, the gases. But another students will analyze this with other thesis.
Just to clarify, when Fernanda is going to come to Argentina, and I hope that to be soon, once this COVID pandemic finishes, she will going to be measuring the emissions going out from the soil. So the pecan growth is there, or the pecan trees are there only as like a sort of, or disturb of the environment of that pasture growing there.
I'm sorry, I didn't quite get that. Can you repeat that, Sebastian?
That we have a plot with pecan growing there. As I said in the chat, they are 7 years old plants. We have those plants in under the framework of a bigger national network for pecan. And that soil, except for the pecan planting, hasn't been disturbed for 7 years. We have already characterized the soil chemically and physically. The idea is, well, to work on those parameters, on soil parameters, and not that much in the tree parameters. Of course, that we are taking measurements of the tree growing there, mostly on phenology of the tree, but as part of Fernanda's training, we are focusing on how to do the sampling, how to install the chambers, what is the right place, and so on.
Yes, but this is, yeah, go ahead, Todd, please. Yeah.
I'm just asking if Gire is there.
Yeah, I'm here. Can you hear me?
Yes, my apologies.
Yeah.
I was muted.
Okay. Can you hear me now? Okay. Regarding the sampling method that was earlier discussed, I think changing the volume is going to matter because of the flux. The more the volume, the less the flux. So I think what we normally do is that we get a place adjacent to the place of sampling where it is free of grasses, maybe the bare soil, and then we take sample there. And then where we are taking our sample, we consider the biomass and the soil. Then at the end of the day, you now take the other sample from the bare soil, you minus it, you can get the flux from the biomass. But when you start sampling, maybe at the first stage, if you are dealing with like rice, in the first 3 weeks, the rice is, the height is going to be taken care by the volume of a particular volume. But when the plant get taller, and then you now change the volume, I think there is going to be differences in the fluxes from the initial point of start and then toward the end. So if you want to take care of the biomass, that is the plant that is growing in the chamber, you take an adjacent place where it is free of the grasses, you take the sample there, and then you take that of the biomass, and then you minus it, and then you get what you have. But like somebody said, it depends on what you are measuring. Are you taking the emissions, but from the soil and the biomass, or you are taking the fluxes only that is coming from the biomass? So that will determine how best you now go on with your sample. So that's my contribution. Thank you very much.
Thank you. Juan Pablo asked a question in the chat that Juan Pablo, did you want to ask or make your comment to the group?
Hi, it was more like a comment because you were proposing like nobody addressed the topic of biomass in trees, and I'm working on that topic, and actually it's quite difficult to find how we are going to do, how we estimate or measure carbon sequestration. I mean, the PhD is just 4 years, and you cannot go to the field every year to measure the trees and then to try to estimate how much has a tree grown. So we have to rely on aromatric equations, but these are not developed for most of the species, at least in the neotropics. So I was wondering if any of the guys or the people that is participating or you has an experience with these things.
Does anybody have any experience with this that they'd like to share? No one? Saniro? Do you have experience with allometric equations?
Well, not really. I know that
Okay.
there are many of them out there, and when it but when it comes to agroecosystems with different species, it can be challenging indeed. And to be honest, I don't know how to address that.
Yeah.
Using allometric equations.
So largely, people are using mixed species allometric equations in those kinds of situations. It's hard for me to understand whether or not they're less accurate because they're mixed species, or they're less accurate depending on if you're in an agricultural setting, because agricultural trees are not growing in the same way that forest trees are growing, which is where most species allometric equations are coming from. So they actually went into the forest and harvested trees to develop those equations. And so in a lot of ways, it's you can use those mixed species allometric equations depending usually on your sort of climate zone, but it'll at least give you some estimate of biomass and potential biomass gain. And so this is probably the best estimate that you can get without going out there and as you say, measuring every year and doing destructive sampling.
Yeah.
Yeah, thanks.
Yeah.
Can I, well, so, yeah, actually, I just finished a paper and it's almost ready to submit because we are trying to propose an idea how to deal with this problem. Because it's not about estimating the carbon content, it's about estimating the carbon sequestration potential. And of course, if we are in a forest plantation, then it's easy to know how old is the plantation, and then we can try to estimate for how long this tree is going to grow. But when you are in farms, in small farms where there is no data about how old is the tree, or sometimes there is not even information about the species growing type. So I think that is one of the lacks of information.
Yeah, there's very little information of trees on farm. I'd be very keen to see the paper that you're about to put out and hear more about your proposed solution for this.
Okay.
And if I may recommend, there is also a paper published in 2018 from CLIFF's colleague called Diana Feliciano, and I can email you all this paper. So she basically did a literature review on above ground and below ground carbon sequestration in many types of agroforestry systems. And she put together lots of references for doing this review that can, I'm pretty sure that it can help you out trying to figure out ways to measure and to compare different types of agroforestry systems.
Yeah, thanks. Actually, I read that paper and I cited it. So I hope it gets published, then I could share.
Yeah. In the paper that's come out more recently than Diana's paper is the Remy Cardinal paper, which was revising the IPCC estimates for agroforestry in environmental research letters.
Oh, okay, I will check. Because also I'm using drones to try to cover bigger areas. So yeah, that will be the next step.
Yeah. And from the drones, you're trying to correlate tree cover or crown area with biomass? Is that the goal?
Yeah, from the drones you can get tree height and crown diameter. Then you can use an equation to estimate biomass. But
What
again,
kind of sensor do you have on the drone?
no, I am just using RGB, but it's possible with photogrammetry. Ideally, I will be using LiDAR, but yeah.
Yeah, sounds great. Thanks for, this is taking the conversation into a whole different direction, which is important for the integrated systems because there's nitrous oxide happening, there's biomass and everything. So thanks for sharing, Pablo.
Yeah, and Todd, I think our time is up.
Okay.
And again, I thank you so much for taking the time to talking to us and being here with us and share your experience in this field. I also thank all the students that showed up, and I really hope you have enjoyed this session. And as I said, 4 more technical sessions will be out in the next weeks. So please stay tuned. And if you have any doubt, want to follow up on any topic, please do not hesitate to email us, right? We are happy to help. And I bet Todd is also happy to help on his end. And Hazel, would you like to put something before we wrap up?
Just to reiterate what you've said, Saniro, thank you so much to Todd for chairing the session. It's been really, really valuable to all of the students, I'm sure. And I'm also sure that there are probably a lot of questions that were unsaid. So what we will do is we'll have an email chain of everyone who's attended this meeting, and we will send out the resources that have been discussed, and anyone can kind of add any of the publications that have been discussed to that email chain. Yeah. So thank you once again to everyone, and thank you, Saniro, as well.
Thanks
everybody.
So
have a great night, day, wherever you are.
Thank you. Thank you. Bye, Todd. Enjoy the sunshine.
Yeah. Have a good day.